Today we're going to be talking about the molecular basis of breast cancer. And this is a pretty broad topic because there are so many different forms of breast cancer. So we're going to be touching on a few different types and then really focusing in on the mechanism of action of estrogen receptors and how those affect the development of breast cancer. So let's just jump right into the signs and symptoms. Some of these may be able to be seen from the outside and others may not be able to. So one of the most common symptoms of breast cancer would be a lump in the breast, and this may or may not be able to be seen from the outside. Other symptoms would be a change in the skin, like redness or swelling, a change in the size or shape of the breast, as well as inverted nipple or discharge from the nipple. And then there also may be issues with the lymph nodes, and the lymph nodes in the breast or armpit area may be swollen. There are two different models of breast cancer initiation and progression. The first is the clonal evolution hypothesis, and the second is the cancer stem cell hypothesis. So the clonal evolution hypothesis states that any cell susceptible to sporadic random mutation and a particular combination of mutations allows selection of a cell to evolve to become immortal and tumorigenic. Thus, any cell within a tumor can maintain tumorigenesis. So basically, any cell in our body that becomes mutated can then further divide and create more mutant cells. The cancer stem cell hypothesis, however, states that only stem and progenitor cells, which are a minor fraction of cells within a tumor, can give rise to self-renewing tumor cells. So stem cells are the cells that create all of the different types of cells in our body. Progenitor cells are a little bit more specified, so they can only create a few different types of cells, not any cell like a stem cell can. And these two types of cells only take up a very small portion of all of the cells that are found in a tumor. Most likely, the reality of the situation is that what happens in a tumor is a combination of both of these hypotheses. Breast cancers are categorized by 4 different criteria: the tissue in which they arise, their histology, their hormone sensitivity, as well as their genetic makeup. So we have 3 different types of tissues that breast cancers can arise from: the ducts, the lobules, and the connective tissues. So in the ducts and the lobules, it's either called ductal carcinoma or lobular carcinoma, depending on where it starts. And then if the cancer arises out of the connective tissue, it's called sarcoma. So generally, within the ducts or the lobules, the cancer will start inside of the structure, and then once it moves outside of the structure to surround it, it's called invasive ductal or lobular carcinoma. Carcinomas make up the majority of breast cancer cases, and these cancers arise from the epithelial cells that line the ducts and the lobules. These cells are what normally produce milk. Sarcomas arise from connective tissues like myofibroblasts and blood vessel cells. These cases make up less than 1% of all breast cancer cases. There have been many different genes associated with different types of breast cancer. BRCA1 and BRCA2 are the most commonly associated genes with breast cancers. And these 2 genes play a major role in the DNA repair mechanism. We know that 5 to 10% of all breast cancer cases have a hereditary component, and also that familial breast cancers have a malfunction in the DNA repair mechanism. Many different breast cancers are hormone sensitive, so we have 3 different receptors that we're going to talk about: the estrogen receptor, the progesterone receptor, and the human epidermal growth factor receptor 2, or HER2. Estrogen receptors are ligand-inducible transcription factors that regulate many different genes. Estrogen receptor positive cancers have a better outcome than estrogen receptor negative cancers because they can be treated with targeted therapies. Estrogen receptors are important in breast cancer, but so are progesterone receptors. If the progesterone receptor isn't working, then it shows a lack of estrogen receptor function and thus a worse prognosis for that type of breast cancer. 1/4 of all breast cancer patients have a malfunctioning HER2 gene. 20% of invasive ductal carcinoma patients have an overexpression of HER2. So what is this? It's a protein that is found in normal, healthy breast tissue that functions to help the cells grow, divide, and repair. However, if there's an overexpression of this protein, then the cells will grow and divide out of control, and this is a hallmark of cancer. So HER2 positive breast cancer will tend to grow more quickly, spread to other areas of the body more quickly, as well as have a higher likelihood of recurrence versus HER2 negative breast cancers. While we're talking about hormone-sensitive breast cancers, I feel like I should mention triple-negative breast cancer. It does not express estrogen receptors, progesterone receptors, or the HER2 protein. And these breast cancers are known for a very poor outcome because there aren't targeted therapies for them. They don't have any of the receptors that the other breast cancers have. Now we're going to get into the mechanism of action of the estrogen receptors. There are two different receptors found in our bodies, ER alpha and ER beta. They are coded for by the ESR1 and ESR2 genes respectively. Estrogen receptor alpha is the receptor that regulates normal breast development as well as breast cancer. ER alpha has a high affinity of binding to estradiol. Estrogens are a group of steroid hormones that are found in the human body. The three most common estrogens that are found in the female body are estrone, estradiol, and estriol, with estradiol being the predominant one found during reproductive years. Estrogens are necessary for proper breast development. However, if too much estrogen is present, it can cause DNA damage, which causes cancerous cells to form. How does that DNA damage happen, though? Estrogen activates many genes by transcription. So when the RNA is transcribed, sometimes it can stick to the DNA, creating an R loop. This R loop causes the DNA to break and causes changes in the genome, which make the cell cancerous. So, an increased level of estrogen means an increase in R loops, and thus an increase in DNA breaks, and an increase in cancerous cells. These DNA breaks are found particularly in regions of the genome where estrogen-activated genes are found. As you might guess, since estrogen binding and receptors are associated with DNA transcription, they are found in the nucleus of cells. There are a couple different mechanisms of action in which the estrogen receptor uses, and so we will talk about all three of them. The first is the classical mechanism of estrogen receptors, and this response occurs in a matter of hours. So in this one, estrogens will diffuse into the cell and bind to the estrogen receptor that is found in the nucleus. And you can see in this diagram that the little purple estrogen at the top goes down towards the left arrow. This is mechanism 1, and will bind to the estrogen receptor. This then forms the estrogen receptor complex, and this complex can then bind to estrogen response element, or ERE, sequences. And this is done through protein-protein interactions with activator protein 1, or SP1 sites, in the promoter region of estrogen-responsive genes. And this binding takes place in order to regulate the transcription of target genes within the nucleus. Once bound to these genes, then co-regulatory proteins can be recruited to the promoter, and mRNA production can be either increased or decreased. The next mechanism we'll talk about is non-genomic mechanisms, and this is number 2 in this diagram, and this will be a quicker response, so the response will happen in seconds to minutes instead of hours, like the previous mechanism we talked about. The estrogen receptor will also be located in or adjacent to the plasma membrane instead of in the nucleus, like the classical mechanism. The estrogen receptor is sent to the plasma membrane by adaptor proteins like caveolin-1 or SHC. Binding of estrogen to the receptor causes a rapid change in cellular signaling molecules and a stimulation of kinase activity, and this will increase the levels of calcium ions or nitric oxide. The last mechanism will be non-ER plasma membrane associated estrogen binding proteins, and that is mechanism number 3 in this diagram. And this happens through a signaling cascade when estrogen binding proteins may trigger an intracellular response. There are 2 hypotheses as to why excess estrogen causes breast cancer. The first of these is that byproducts of estrogen metabolism directly cause damage to the DNA, thus creating more point mutations and cancerous cells. The second is that when estrogens bind to the receptor, it causes the proliferation of mammary cells, and this will increase cell division as well as DNA synthesis, and an increase in these elevates the risk for replication errors and thus causes cancerous cells. So how is breast cancer diagnosed? Well, our first line of defense is just a self-breast exam. Doing that frequently causes a lot of women to be able to catch abnormalities in their breasts early on, and thus gives them a better prognosis. The testing method that is used most frequently is mammograms, which are an X-ray of the breasts. And in this mammogram, you can see that the diagram of how the X-rays are pointed down at the breast that is squished between two plastic trays. And this compression is to flatten it out to make the X-ray a little clearer. You can see 4 different images here. We have a normal mammogram on the top left showing normal breast tissue, and then the top right shows a little cancerous lesion that is pointed to by the arrow. You can see also the benign cyst on the bottom, as well as just calcium deposits. So there are many different readings for these mammograms that could occur. If something suspicious is detected in a mammogram, generally either an ultrasound or an MRI will be ordered. You can see in this ultrasound that there is a dark mass, which the arrows are pointing to in every image, and that shows a cancerous mass. And then I also have the MRI image, which shows a normal breast tissue on the left, as well as cancerous areas, which are those white dots in the circle on the right side. Once this imaging has been done, a biopsy will most likely be ordered of the suspicious tissue. When the biopsy is done, hormone receptor tests and HER2 tests will be ordered to help guide the doctor in the treatment. If the tumor is estrogen receptor positive, it means the cells have receptors for estrogen, and this is about 2/3 of breast cancer cases. If it is progesterone receptor positive, it has receptors for progesterone, and progesterone could help promote the growth of this cancer. The tests that look for the HER2 proteins can be done by biopsy or by a serum test. And these are looking for 2 different things. Is there excess HER2 in the cells? And do the cells contain too many copies of the HER2 gene? There are many different treatments approved for the treatment of breast cancer. Generally, the first step in treating breast cancer is to do either a lumpectomy or a mastectomy, depending on the size of the tumor. A lumpectomy will be done for a smaller tumor, and the goal of this surgery is to preserve as much breast tissue as possible. So the tumor will be taken out, as well as some of the surrounding healthy cells, so to make sure that all of the cancerous cells have been taken out. You don't want to leave any in there. Generally, after a lumpectomy, radiation will be done, and this is to ensure that all of the cancerous cells have been killed and or removed. So radiation is usually done through a machine that shoots X-rays or protons into the body to kill these cancerous cells. And this is done through a machine that rotates around the body. Sometimes probes will be stuck into the breast to cause radiation from the inside out, but most of the time it is external radiation that is done for this cancer. Hormone therapy can be used for up to 5 years in conjunction with other therapies like a lumpectomy and or radiation. In postmenopausal women, hormone therapy can be used in conjunction with aromatase inhibitors, which inhibit the amount of estrogen that the body produces. The National Cancer Institute has a long list of medications that are approved for the treatment of breast cancers. I'm going to be focusing in on 2 common medications that are used, but if you would like to see this full list, you can go to the National Cancer Institute's website, which I will have in my sources page as well as linked in the description below. Tamoxifen is commonly used to treat breast cancers, and it can be an ER antagonist or an ER agonist depending on which tissue it's in. So in bones and uterus cells, the tamoxifen is an ER agonist, whereas in breast cells, it is an antagonist. So it's used as an adjuvant endocrine therapy for breast cancers. The active ingredient in tamoxifen is a triphenylethylene derivative, which will bind to the estrogen receptor instead of estrogen. And the suggested mechanism of action for this triphenylethylene derivative is that it binds to the ER and then it helps to produce transforming growth factor beta, or TGF beta. And TGF beta is a cytokine that is able to regulate cell growth, differentiation, and apoptosis. So if this cytokine is increased, then cell growth is able to be better regulated and fewer cells are mutated because they're not differentiating as quickly, as well as cancerous cells can be killed. It has been shown that in 5 to 10% of breast cancers that are ER negative, tamoxifen is still effective. So there must be another mechanism other than the estrogen receptor mechanism that we know of, but that is still being researched. Raloxifene is an approved medication for the prevention of breast cancer, and similarly to tamoxifen, it is a tissue-specific agonist or antagonist for the estrogen receptors. And in the bone and uterus, it will be an estrogen receptor agonist, and is actually used quite frequently for the treatment of osteoporosis. The active compound in raloxifene is benzothiophene, and that is the compound that will bind to the estrogen receptor. How can a medication be both an agonist and an antagonist, depending on what tissue it's in? We have these things called selective estrogen receptor modulators, or SERMs, that bind to the estrogen receptors and cause a conformational change in the receptor. This conformational change determines whether coactivators or corepressors will bind. So the relative levels of coactivators to corepressors in a tissue determine the activity of the medication. For example, tamoxifen recruits a coactivator to estrogen receptor genes in endometrial cells. However, it recruits a corepressor to these same genes in breast cancer cells. The relative levels of coactivators and corepressors play a role in the activity of the medication, but also the relative ratio of estrogen receptor alpha to estrogen receptor beta in a tissue make a difference. So whichever one predominates will determine whether the medication will be an antagonist or an agonist in the tissue. I mentioned aromatase inhibitors earlier, and these are medications that are able to block an enzyme, aromatase, from turning androgens into estrogens. And the reason that these medications are used is so that there is a reduced level of estrogen in the body. If the receptor doesn't have any estrogen to bind to, then it can't produce its function. A slightly more controversial treatment option would be using mushrooms to treat breast cancer. There was a study published in 2018 that said that mushrooms may be a good new source of treatment for breast cancers. And I would argue that it's not a new treatment at all, because mushrooms have been used for thousands of years in traditional Chinese medicines, as well as traditional medicines in other countries. But it would be newly brought to research as well as to America. This study specifically focused on 4 different types of mushrooms, but I'm only going to talk about 2 of them, the chaga mushroom and the turkey tail mushroom. And these 2 are the mushrooms that are specifically pertinent to breast cancer cases. Chaga mushrooms grow on birch trees in cold northern climates. They have been used in Russia, Japan, China, and Korea as a cancer treatment. They have been found in recent studies to have immune regulatory effects as well as many cytotoxic effects. They have been tested on both HeLa cells and mouse models. Many different compounds in the chaga mushroom create these effects, including growth arrest, apoptosis, and immunostimulation. The turkey tail mushroom grows in many climates throughout the world and has been found to have immunomodulatory effects. It has been used, a specific compound in this mushroom has been used in Japan since 1977 and in China since 1987. Beta-glucan-based polysaccharopeptide fraction, or PSP, and polysaccharide fraction crestin, PSK, are 2 compounds that have both shown the best immunomodulatory effects. It is PSP that has been used in Japan and China. PSP is able to activate cells of the immune system as well as boost production of cytokines and chemokines like tumor necrosis factor alpha, interleukins, histamine, and prostaglandin E. It is also able to stimulate dendritic and T cell infiltration into tumors and reduce the harmful side effects of chemotherapy. So in many Asian countries, mushrooms are used along with chemotherapy to treat cancer, not on their own. And that is all I have about breast cancer for today. I will have all my sources here on this page, as well as I will link them in the description below.